All right, so this is our Lab2 overview. Again, you can see the tasks, you have the homework module, same thing, the lab pages that you have to submit, and the quiz that is also due on the 22nd. So these first two labs are a little tricky because they're not as based in structures, they're more conceptually based. So having to learn the hormones, their functions, their release, their stimuli, and all of those things. Same thing for this lab. This lab is just covering the blood. So it's not covering vessels, it's not covering the heart, that's chapters 19 and 20. This is just talking about the blood and specifically the formed elements within the blood. So the different types of cells, your red blood cells, your platelets, and your white blood cells. So that's what we're gonna be looking at this lab, as well as blood typing. So the different blood types A, B, and O, how transfusions not necessarily happen, but I'm looking at the antigens, and so issues for transfusion and factors to consider, as well as for genetic inheritance of blood types. So for this chapter, or for this lab, I really highly recommend you use your book. It's gonna be very helpful for this. There's not as much stuff in the APR itself for this one. Maybe in the cardiovascular overview, there's definitely some really good videos here in the cardiovascular overview. But as far as blood, the book is going to be more helpful for this, I think. So looking at your lab, identifying the blood cells, so whole blood, erythrocytes, platelets, basophils, eosinophils, lymphocytes, monocytes, neutrophils, and identifying sickle cells, so cells that are diseased with sickle cell, as well as a leukemia slide. This one, apparently you're finding online. So you should be able to identify them in your textbook and on micrograph images, interpret blood typing data to determine a person's blood type, explain how antigens and antibodies interact in the blood, and what happens if you mix blood types. So it gives you this very helpful table where it tells you the different blood types, both their phenotypes, so A, B, and O, as well as the positive and negative, and what their genotypes look like. So this is where I usually remind people that if your Mendelian genetics is a little bit weak, if you can't quite remember Punnett squares, homozygous, heterozygous, dominant, recessive, stuff like that, take a break, pause this video, and go watch something on Crash Course from YouTube or Amoeba Sisters, some type of genetics, intro to genetics video, just to help you remember what's the difference between a genotype and a phenotype. What is the difference between a recessive and a dominant allele? How are things inherited in that sense? Because we're going to be working with, for this chapter, it's very straightforward genetic stuff, but if you don't have that background, you're going to get lost really quick. So if you haven't figured that out yet, if you don't have that stuff, maybe go ahead and pause this and find that first before we go any further. Okay, let's look right in here. So again, your book, when you take blood and you centrifuge it out, so if you ever go and donate blood, they take it right out of your arm, they put it in a tube, and they send it off to the lab. Lab is going to centrifuge your blood. It's going to spin it out and separate out the components, because there are three different components that actually make up your blood. Those are the plasma, the buffy coat, and the erythrocytes. So the plasma is the fluid portion of blood, it's the water portion of blood. It also contains the proteins and specifically the antibodies. And that is an important thing to remember when we get into blood transfusions later, but it's the plasma that contains the antibodies. The red blood cells are the heaviest layer. They sit down here at the bottom. A lot of times if you are donating blood or if you're receiving blood, you're receiving red blood cells. You're not actually receiving the plasma. You don't receive whole blood typically. You typically receive just the erythrocytes from somebody. And so it's that, again, we'll talk about that when we get into blood transfusions, but the plasma has the antibodies. Then you have the buffy coat. That's going to be your white blood cells, the 5 different types of white blood cells, as well as your platelets. And then again, red blood cells down at the bottom. If we go in here, so this is 18.3 formed elements in the blood. It gives you that whole slideshow about how these formed elements actually occur. But down here towards the bottom, there's a very helpful picture. I know we're scrolling right past the blood transfusion, blood stuff. Here we go, table 18.7. This is the table of your leukocytes. So this is a very, very helpful table in identifying the different types of leukocytes. It divides them up between the granulocytes and the agranulocytes, which this may shock you, but the granulocytes have tiny little visible granules in their cytosol, while the agranulocytes do not. So there's your first kind of how are you going to identify these cells? If you were just given a picture of a cell like you have in your lab manual, how do you identify it? Well, the first thing is it a red blood cell? Because those are easy to identify. You'll see those in here. It's all of these that are floating around. Is it all of these tiny little cells? Okay, if so, it's a red blood cell. If it's something else, the first question you should ask yourself is, well, are there granules visible? Are there little dots in the cytosol? Or is the cytosol very kind of clear and smooth looking? If it's clear and smooth looking, then you can say, okay, it's an agranulocyte. If there is cytosol, if there is these little granules, then you say, okay, well, it's a granulocyte then. And what they use those granules for are to actually, they release them. Those are their chemicals that they will release to go after pathogens. And the agranulocytes technically do have granules in their cytosol. They're just so small you can't see them, so they appear to be agranulocytes. I know that's lovely and confusing. But again, this chart is going to give you pretty much all the detail that you need. So you have neutrophils. The nucleus is multi-lobed. Cytosol contains neutral or pale granules. So if we go up here and we look at the neutrophils, you can again see this kind of multi-lobed nucleus. It's not nice and smooth. It has all of these different kind of bulgy sections in it. It has these granules that you can kind of see. Some of them pick up stain, but really they're just kind of there. They're just kind of dots that are sitting in there. So that's that neutral pale specific granules that it has inside of its cytosol. If we look at something like a monocyte, so monocytes, they have kidney-shaped or C-shaped nucleus. Usually it's pale staining, abundant cytosol around the nucleus. So those are some features that we can look at, and we can see, okay, we've got this kind of kidney-shaped, C-shaped nucleus. It's not as dark as some of the other cells, how they stain, and you have this abundant cytosol all around it. Okay, this one, these lymphocytes, this is very characteristic, having this super dark staining nucleus that fills up almost all of the cell, very characteristic of a lymphocyte. So this is again very helpful in identifying different cell types. If you go into here, again, you have all of these red blood cells, these erythrocytes. That's what's all floating around. That's kind of what this thing is pointing at right there, is an erythrocyte. But you see, you also have other types of cells. So you can identify all the cells in here. If I were going to do that, I would say, well, I have some erythrocytes. I have these red blood cells. There are some neutrophils. So, let me see this. Let me get a little bit closer. So down here towards the bottom, you've got a neutrophil. That's a neutrophil. That one up there. This one down here, so you have two neutrophils right here, and this one below it, I would call that a basophil. I would call that either a basophil or a lymphocyte. It almost looks like, so it's not a great picture, so it looks like there's some kind of color differentiation between the nucleus and the cytosol, so I think that one might be a basophil. Same thing with that one up there in the corner. But these little fragments, you should see these little fragments in all of the pictures. In those, as well as in some of these other ones, these little kind of specks that stain. Those aren't mistakes, those are platelets. Okay, so you have erythrocytes, you have the little dark dots, which are platelets, and these mostly are neutrophils. This one, I would call that definitely a monocyte. You've got this good C-shaped nucleus, lots of abundant cytosol around it, no granules, looks very clear cytosol. So that one would be a good monocyte. You've got these over here. So again, down here, looks like a little basophil, some red blood cells, lots of platelets. Then you've got these three cells that are all the same type. So if you're like, okay, well, what kind of cell is that? Let's go look. It's, first of all, has these specks in its cytosol, so it has granules, so it is a granulocyte. So we can say, okay, so of the granulocytes, is it a neutrophil, an eosinophil, or a basophil? Well, we can look pretty quickly and see that looks very much like an eosinophil to me. So if I go down here, eosinophil nucleus is bilobed, contains reddish or pink orange granules. Okay, well, yes, we have a bilobed nucleus. You have one kind of bulge, a little thin strip, and then another bulge. Same thing here, kind of these two bilobed nuclei, and kind of pinkish orange looking staining specific granules. So yeah, that would be an eosinophil. Oh, it also doesn't hurt that it tells you right there, does it? There you go for copyright purposes. But then there's a couple more that you can practice with in identifying the different cells. This one, it's asking you to circle two irregularly shaped and describe the diseases that would lead to this altered shape. So you should be able to identify within this slide the cells that are normal, the cells that are not so normal, and what would cause that. These are just, again, good practice questions. Once more, if you are looking for ways to study, here you go. Here are ways that you can study. Okay, so this chart, this is usually a wonderful headache for students, but don't worry, it won't be for you because we are going to walk through half of it together, so don't worry. Once we do the chart though, the pages that you have to submit are the critical thinking questions and the practice problems for inheritance. So we're going to go through all of this stuff, and then you have to submit these practice problems for transfusions and inheritance. It looks like a lot. You don't have to do crosswords. It looks like a lot, I know, but once you start working with it, it really is not that bad. Some of them are yes/no questions. Some of them are just very straightforward. Repeating the same questions for a slightly different blood type. So with blood typing, there's a pattern that starts to develop. Okay, so let's go over to the board. It's very crooked. Okay, so looking at this chart on page 24, reviewing some just basic terms to start with, antigens and antibodies. Antigens are what is on the cell's surface, and that is what identifies it as a healthy cell, an infected cell, a cell that's native to your body, or a foreign cell. So for coronavirus, the antigens that it has on its cell surface are these spike proteins. So coronaviruses all have these spike proteins. And so it has these antigens that are like spikes that are all on its cell surface that antibodies can bind to. Or the antigens can bind to other cells in our body and infect them. So when we talk about antigens, every cell in your body has antigens on its surface to identify it as, again, a healthy cell, a body cell, or a foreign cell, whatever it may be. I always think about like kind of ships, like flags on ships, kind of flying their colors and saying and identifying themselves. So starting with blood type A, positive. A positive. We have our antigens. So the antigens for A positive blood are both the A antigen and the RH antigen. So if someone has A positive blood, that means that their red blood cell is going to have little A flags and little RH plus flags all around its cell surface saying, hey, here's who I am, here's what I am. If someone has type A blood, they will have, and here's how we write this, anti-B antibodies. So someone who has type A blood has type A positive blood has A antigens and RH antigens on their red blood cells. Their plasma, so the fluid portion of their blood, is going to have anti-B antibodies. So if you have type A blood, you cannot receive type B blood because you have antibodies for that. Okay? So this type can donate to A positive and AB positive. So same can always give the same. So the A positive will always give to A positive, B positive to B positive, O negative to O negative. You can always give to yourself. A positive can also give to AB positive. And that's because we'll see that AB positive blood does not have any antibodies. And in fact, AB positive is what's called the universal recipient. So they can receive any type of blood. A positive, O negative, B negative, AB negative, doesn't matter what it is. So in this donate to column, this type can donate to, AB positive should be in every single one of those. Okay, because it does not have any antibodies in its plasma. Right, receive from. So A positive blood, you can receive from yourself. If you're positive, you can receive from a negative. And O positive negative. So I'll go through that. You can always donate to and receive from yourself. So A positive can receive from A positive. A positive can also receive from A negative. So the general rule is that positive can give to positive, and negative can give to positive or negative, but positive cannot give to negative. Okay? So positive to positive, negative to positive or negative, but positive cannot give to negative. So in this case, if you have A positive blood, you can receive from A positive or A negative, as well as O positive or O negative. Now, just like AB positive was considered the universal recipient, and you should have AB positive in all of your donate to column, you should have O negative in all of your receive from column, because O negative is the universal donor. Okay, so O negative has, I know I said just a few minutes ago that all cells have antigens on their surface, except for O negative red blood cells. They don't have any of these antigens on their surface to identify themselves. And since they don't have any antigens, it doesn't matter how many antibodies you have, they won't be able to recognize that cell. There's nothing on that cell surface for them to bind to. And so you can give O negative blood to anybody, any blood type that they have. You cannot give O positive blood to any blood type because, again, positive can't give to negative. So that's a little trick that you have to make sure you're looking at. Is it positive, is it negative? Who's the donor, who's the recipient? When we talk about blood transfusions, I haven't said this yet, I'm sorry. When we talk about blood transfusions, you have to keep it straight in your mind. Who is the donor and who is the recipient? Who is getting the blood, who is giving their blood? Because again, if you're receiving somebody's blood, that means that you have your plasma, your antibodies still in you. But if someone is donating, they don't have any antibodies. They're only giving their red blood cells. So it's not a factor in that sense. That's why you can have positive to positive, negative to positive, negative, but not positive to negative, because that will cause an agglutination. Okay. The last one is the all possible genotypes. So you probably have to go back to your lab manual for this, that very first page. It had the different genotypes listed. So if the phenotype is A positive, the possible genotypes, here's how we write this out. IA, little I. So this takes care of that A. So when we're talking about A positive blood, we have to list out the A antigen, or the A factor and the RH factor separately, because they're inherited separately. You don't automatically get the A and the positive from a parent. You get the A, and you may or may not get the positive. Those two things are inherited separately. So this takes care of our A. We can either be homozygous for A, where you have big IA, big IA. This big I, or these I's, by the way, stand for immunoglobulin. So IgA, immunoglobulin A, right? So IA, or IA, little I. This little I is the recessive allele, and it's for the O blood type. So you have A is IA, B I, B, and O is little I. So A and B are codominant, and O is recessive. So if you have A positive blood, you could be homozygous for the A, IA, or you could be heterozygous for A, IA little I, and still show A. But you would be a carrier for this O allele. For the Rh factor, the positive, you can either be big D, or big D, little d. And that's because positive is big D, negative is little d. So positive is a dominant trait. You can be homozygous or heterozygous for it and still express that allele. So antigens, antibodies, donate to, receive from, and the genotypes. You should have all of these things filled out in your boxes. Okay, let's do B negative. So I'm gonna go through this one just as fast. B negative. So again, you are talking about red blood cells with B negative antigens on their surface, meaning they have the B, the ABO, the B, but if they're negative, that means they have no Rh factor. They have no Rh antigens on their surface. So the antigens that we'll list are just B. This is the negative, so there's no Rh. The antibodies that this person has, so they have B blood, meaning they have anti-A antibodies. Okay. So some people might be tempted to say, well, they're negative. Well, sorry, let's say this. So if A positive has A antigens and Rh antigens, and B negative only has B antigens, shouldn't B negative blood have anti-A antibodies because B can't receive from A? But this is negative, so shouldn't it have antibodies for the positive? And some people might be kind of tempted to say, well, they have what would be considered anti-D antibodies. Because that's again, we're talking about D. So Rh factor, the D antigen is how we call the antigen. So some people might say, well, this should have anti-D antibodies then. But that's only the case if they've been exposed. So if exposed to Rh positive blood. So if you have B negative blood, you are going to naturally, just through your natural body processes, have anti-A antibodies. Those are just going to be part of your body chemistry, your blood chemistry. What won't be are going to be the anti-D antibodies for that positive feature. So what that means is that if somebody is given an Rh positive blood, so say somebody's B negative and they're given accidentally B positive blood, it doesn't mean that their blood will immediately attack that Rh positive blood, because there won't be any antibodies in the recipient's blood to begin with. So if you have B negative and you're getting B positive, it won't immediately react, but over time your body will start to reject it because it will start to develop antibodies to that positive factor. Where we see this a lot, where we see this not a lot, but where it becomes a concern is during pregnancy and during delivery. So during delivery, there is some mixing of fetal and maternal blood. And so if you are a mother with B negative blood and your infant has B positive blood, there is potential that there will be some slight mixing, and the fetal B positive blood will get into the maternal system, and it won't be enough to cause a massive systemic agglutination and death or anything, but it will be enough to expose her blood to those positives, to the B positive factor, which would then cause her body to make antibodies against it. This would be fine, again, as long as she never received any positive blood, because if she received positive blood, boom, she has the antibodies, she would die. So what doctors typically do is give an injection to suppress the immune response, so that way if a Rh negative mother is exposed to her Rh positive baby's blood, she does not produce antibodies against that. Because it's really important for the second pregnancy. So if her next pregnancy, let's say the infant was also Rh negative, there's the potential that her body could attack the infant's antibodies, or the infant's cells with those D antibodies. So, when you write this out, if you put just anti-A antibodies, that would be correct. If you put anti-D antibodies, you need to add a caveat that if exposed to Rh positive blood. Otherwise, you should just leave it off there. Okay? So B antigens, anti-A antibodies, B negative blood. So remember, negative can donate to. Who can B negative donate to? You can always give to yourself. So B negative. Remember I said negative can give to positive. So you can do that. It can also give to AB negative and positive. It can receive from, so remember you can go negative to positive to negative, but not positive to negative. So it's only going to be able to receive from B negative and O negative. Again, notice you have AB positive, O negative, O negative. Okay? Well, we're going to dip down a little bit. The genotypes, IB, 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 I. This is a negative. Negative indicates the recessive alleles, so you have to be homozygous for the recessive allele to show it as a phenotype. So little d, little d. Yeah. So hopefully you start to see that there is a pattern that kind of comes up the more and more you do these. And again, I highly encourage you to go through the entire chart. Email your chart to me. I will correct it for you. I will offer you some guidance if you're getting stuck in it. But hopefully you start to recognize the pattern that develops. And spending time doing this will, again, make the practice and critical thinking questions infinitely easier. If you do not have this chart filled out, when you go to do the practice problems for transfusions and inheritance, you're going to be so much more confused. You will have such a harder time with that. So do that chart to begin with. Also, in some of the critical thinking questions, it asks you to do like one, it'll say, you know, pick a blood type and list all of these things out. And then it'll say, do it with all of the other blood types too. You don't have to. For the critical thinking questions, for the practice questions, if you do it with one blood type, that's fine. Go on to the next question. Yeah, I think you should be okay going through these. Oh, let's do an inheritance problem. Okay, so I'm looking at page 27. So here you go, you're gonna get some answers for these pages you have to submit. So page 27, I'm skipping number 1 because I don't wanna do that. You need to do it too. Number 2, Susie, blood type AB negative, had a baby with blood type A positive. Could Bert be the father? Blood type B positive. Why or why not? So Susie is AB negative. Bert is B positive, and their baby is A positive. Okay. Could Bert be the father? Why or why not? How do you start to figure this out? So to figure these kinds of questions out, we have to look at the genotypes. So Susie's genotype, IA, IB, little d, little d. So we have the AB and the negative. Bert, he could be IB, IB, or IB, little I. And then since he's positive, we don't know. He could be big D, or big D, little d. He could be either one of those. For the baby could be IA, IA, or he could be IA, little I. He's positive, so it could be big D, or big D, little d. So this is how you start to figure out these types of inheritance problems, is that you have to look at the genotypes and list them out. And really what you're trying to say is, okay, we know that Susie's the mother. We need to try and figure out, does Bert have the genes to be this baby's father? So if the baby is A positive, we know that it's going to get the IA from Susie. So it gets the IA from Susie. If Susie is negative, she has RH negative, she has little d. So then we know that she's going to get a little d. So we know then that the baby is big D, little d. So here we can say, kind of just, this is kind of like a math problem almost. We're simplifying it down a little bit. So for baby, we can then say, okay, so he's got a big D, little d. So he needs to get a big D from Bert. Well, okay, yeah, Bert has, or let me rephrase that. The baby would need to get a big D from their father. And so Bert has big Ds, so yes, he passes that test. Okay, so in that sense at least, yes, Bert could be the father. Now the second thing is that the baby is A. If we look at these, we know that Susie is going to give an A, so we know that it's at least IA. What we don't know is what is this second allele? What is that second allele that's going to go in there? Is it going to be another IA, or is it going to be a little I? It could be either of those. If we're looking for another IA, Bert couldn't do that. You know, he's got IB, just not going to work. But if we look for a little I, Bert has that. He does have a little I. Remember, you only get one allele from each parent. So the baby will get their IA allele from Susie and a little d from Susie. They'll get this little I from Bert and one of these big D's from Bert. And what do you know? Baby's not going to be IA, because if Bert is the father, they're going to be IA little I and heterozygous positive. And there you go. You have A positive. Could Bert be the father? Yes. The why or why not, your explanation, is your work. Okay? If you want to do a Punnett Square with it, you can take a Punnett Square and do Susie. You can show Bert over here, and you can do, you know, the full Punnett Squares. This is a good practice for yourself anyway, because we will come into genetics towards the end of the course. But you know, that could be, that's part of the justification. You still have to do the positive though. So you have to do another Punnett Square to do the positive, unless you want to do like a whole dihybrid cross, which I'm not going to make you do. So that's how you would answer number 2. Give me the work, tell me a yes or no. So number 3. I'm going to do this whole page. Could Betty Sue, O negative, have a child who is A positive? So BS, Betty Sue, O negative, have a child that was A positive. Why or why not? So again, we have to look at their genetics. O negative, the genotype, is little I, little d. For C, for the child. So again, we're assuming here that Betty Sue is the mother. We're assuming that she would be. So we can just go ahead and skip the IA, because we know, well, little I, that's still going to be, that's still A. That is just as much type A phenotype as IA would be. So still type A, okay? And then the positive, well, we know that you can, positive is a dominant, so we know that you can be capital D, little d. So here we have Betty Sue, who is what we can call double recessive. Double recessive. That's how you spell recessive. Meaning that she has the O allele and the negative alleles. Her child is A positive, so not double recessive at all. Heterozygous for both. So in this case, if Betty Sue, let's say she found a parent that has A, B positive blood. Well, that parent would have IA, IB, and then some variation of big D, little d, positive genotype. So it doesn't really matter because we're just trying to see, could Betty Sue be the mother? Not necessarily what the father's genotype would have to be, but could Betty Sue have a positive child? And yeah, I mean, any Punnett Square that you do that has the two recessive I alleles, those are going to be masked by any A or B. So if you have an IA, IB, you still have a 50% chance of having a child with type A blood. So could Betty Sue be the mother? Yes. O negative is double recessive. Okay, there's your work. All of that. All right. Again, if this is a lot for you, I recommend find a good video about genetics. 4, could Delphia, O negative, have a child who was A positive, why or why not? O negative, a child, so a child is A, B positive. So Delphia, II, little d, child. If they're AB, they have to be IA, IB. It's the only genotype for AB blood because A and B are co-dominant. And then positive, we could be either way. So we're just gonna go, we know that positive is dominant, positive can be heterozygous and still be showing. We're just gonna go ahead and say, sure. She would only give a little d anyway. So this one we can look at straightforward. So the child will have to get an allele from their parent. If the child is AB positive, the child is AB blood, neither of these alleles are going to work, right? So could Delphia be the parent? No. She only has recessive alleles. She has no A or B allele to give. However you want to say that, whatever words sound good to you. Okay? Last one. So number 5, Josie slept with 3 men near the same time she conceived her son, Jeffrey. We do not shame Josie. We are glad that she is living her life. Josie is A negative. Jeffrey is O positive. Which of the following men could be Jeffrey's dad? Explain why each man could or could not be the father. So again, we do genotypes. IA. Okay, here you go. You have to think ahead. You know Josie's the mother. You already got that one. You know that Jeffrey is O positive. So we can do some deduction about what Josie's genotype is based off of Jeffrey's phenotype. Okay? So Jeffrey is going to be little I, and then some type of big D with something else in here. If Josie is the mother and you get an allele from each parent, she has to be IA, I, because Jeffrey has to get one of these each from his parent, from each parent. If Josie is negative, we know she's going to be little d, so that's what she gives to Jeffrey, to her child. So now we know Jeffrey's genotype. That helps us a lot in figuring out who the father is. We also know Josie's phenotype, or genotype. So now that we know both of their genotypes, we can start to work backwards. So look at Teddy. Teddy is AB positive. So Teddy equals AB positive. If we look out the genotypes, IA, IB, and then some type of D thing, it doesn't really matter, as long as there's one big D. So there's your one hint where you can cross people off pretty quickly. If Josie is negative and Jeffrey is positive, he's got to get a big D from his father. So if any of the men are negative, they cannot be the father. So you could go ahead and immediately cross off Jeremiah and Paul. You can just say, nope, sorry, you are not the father. For Teddy, blood type AB. So if we're saying that Jeffrey gets one I from his mother, he's got to get the other I from his father. There is no way that Teddy could be the father because he doesn't give them a little I. For Chris, it's A positive. Blood type A, you could be IA little I, Josie is, why not? And then again, for this one, it's D whatever. So Chris is positive, so yes, we've got the big D coming in. And he's got this little I that we need. Right? So you've got this D, going to that D from that one. Let's see, it's actually this way. Cool. So there's your yes, and you can, again, explain why for each one. Come up with a good explanation for the two negatives. I prefer the explanations to use the genotypes. I think that it is good practice and it makes it be very exact and precise. Okay, so the next page just has more of those similar types of questions, so hopefully that will help to get you through those. Yeah. Like I said, the critical thinking questions, those are going to be a lot about blood transfusions and blood donations. So one more time, that chart, have it filled out, email me if you want me to go over it, and come see me during office hours.